Okay, students, welcome back. So we were studying about the development of Antheridium as I was describing. The male conceptacles has got a cavity-like structures where this cavity-like structures has been embedded over by the conceptacular wall, and attaching to the conceptacular wall there are certain stalk-like structures called as stalk-bearing Antheridium. Each of this Antheridium is oval in shape, carrying biflagellated anthrozoids. These anthrozoids, 64 anthrozoids, can be observed. 64 anthrozoids can be observed in each Antheridium, and this Antheridium has got two wall layer, exocyte and endocyte. And if you observe each of this anthrozoid, it is single celled, has got a single nucleus, and this anterior end is pointed, and its lateral end has got two different kinds of unequal flagella. One is whiplash, other one is stencil. That is a characteristic feature of the phaeophyta. That is brown algae. That is the characteristic feature. So once the Antheridium matures, once the anthrozoids are produced inside this Antheridium, they get detached from this stalk-like structure, and they go and deposit at the osteole, where the exocyte ruptures and produces certain mucilaginous structures. And through this mucilaginous structures, these anthrozoids escape from the male conceptacle, and they reaches the oogonium. That is, they reaches the female conceptacle. By that time, this oogonium, say this is the young oogonium and this is the matured oogonium. As I was mentioning, the young oogonium, that is the young conceptacles, they have got a sessile oogonium. See, this is sessile oogonium without any stalk-like structure initially. And you know, as I was mentioning, this oogonium is made up of three wall layer: exocyte, mesocyte, and endocyte. This exocyte ruptures, and from the mesocyte produces certain gelatinous structure. This gelatinous structures which has been produced from this mesocyte, they germinates or they develop, and they reach the wall of this oogonium. They develop till they reaches the wall of this oogonium. Once it reaches the wall of this oogonium, it pulls this oogonium from where it has been embedded. It pulls and make this oogonium to expose outside this osteole. So now our oogonium has been exposed outside the osteole, and this oogonium has got a gelatinous stalk. That is the characteristic feature of this sargassum that you have to remember. So now the oogonium is ready for the process of fertilization. At the same time, this Antheridium would have been developed. Anthrozoids are produced. These anthrozoids are biflagellated, as I was mentioning. They are biflagellated, and these Antheridium reaches this oogonium. So anthrozoids reaches the oogonium, and later what happens? So they attach, these anthrozoids attach onto the wall of this oogonium. So how it attach? They attach in such a way that its anterior flagella, that is the tensile flagella, tensile kind of flagella, they attach onto the wall of the oogonium, and the whiplash kind of the flagella, they swim in the current of water. They swim in the current of water. That is also the characteristic feature of this group of organism. Okay, later, one of the competent anthrozoid, and they are involved in the process of fertilization, where the wall between the anthrozoid and the oogonium dissolves. The nucleus of this anthrozoid enters inside the oogonium, fuses with this egg cell to form a diploid zygote. Later, what happens to this diploid zygote? This zygote undergoes one transverse division. They undergo one transverse division, the upper cell and the lower cell. The lower cell, if that particular sargassum is is found to be attached onto the process of substratum, they just develop into the attachment disc. The upper cell undergoes many anticlinal and periclinal division to form the mass of cells, where these mass of cells undergoes a process of differentiation, which leads to the development of the entire thallus. So once again, I will repeat, during the process of fertilization, once the oogonium is ready to undergo a process of fertilization, once it gets exposed outside the ostiole, what happens at the same time, anthrozoids from the antheridium also would have developed. They reaches the oogonium, and what they does, they attach onto the wall of the oogonium. They attach onto the wall of this oogonium in such a way that the tensile kind of flagella attach onto the helps in the attachment of this anthrozoids onto the wall, and the whiplash kind of flagella, they just swim in the current of water. Later, the wall of this anthrozoid and the wall of the oogonium dissolve, which leads to the process of plasmogamy and karyogamy to form a diploid zygote. This diploid zygote undergoes a process of division, transverse division, to form upper cell and the lower cell. The lower cell are involved in the formation of attachment disc. Upper cell undergoes many anticlinal, periclinal division to form a mass of cells. Later, these cells gets distinguished into the various the kind of thallus it has to produce to form a mature Sargassum thallus. So this is about the fertilization and the germination of zygote. So to conclude, let us study the life cycle of the Sargassum once again. See, you know, the thallus, the Sargassum is a diploid thallus. It is a diploid thallus. It is found in the form of, it has been attached, few species, many of the species are in the form of floating forms you can find. However, they looks like a angiosperm plant, where they have got a stem-like structure called as central axis or the main axis. These main axis produces various laterals. They are called as leaf-like structures. These leaves-like structures from this axils produces vesicles or air bladders. They help in buoyancy, and from the axil of this leaves-like structure produces the receptacles. The receptacles, if you see the receptacles, they have some cavities. These cavities are called as conceptacles. These conceptacles are of two kinds. One is a female conceptacles, which produces the oogonium, and the male conceptacles produces antheridium. The oogonium, initially they are sessile. Later, upon maturity, they will have a gelatinous stalk-like structure, which helps this oogonium to project outside the ostiole of female conceptacles. The male conceptacles produces antheridium. Antheridium, each of the antheridium, they produces the mucilaginous anthrozoids. Few phycologists, they also call it as sperm, where this anthrozoid reaches the oogonium, diffuses, fertilizes the female gamete, that is the egg, and finally leads to the formation of the diploid zygote. This diploid zygote undergoes transverse division to form upper cell and the lower self. The lower self is involved in the formation of attachment disc, and the upper self develops into the entire thallus. This is the life cycle of the Sargassum. Next, what is the economic importance of this Sargassum? How this Sargassum is important? This Sargassum, as you all know, this is one of the brown algae. The important cell wall component is alginin. Alginic acid is one of an important component, where this alginic acid has been extracted from the Sargassum. That is, most of the brown algae, from most of the brown algae, this particular alginic acid has been extracted, where this alginic acid has got a high economical value. It has been used in many of the industries. This alginic acid is being used in food industries for thickening of this soups, as well as in drinks, in ice creams, thinning of meats, and also it has been used in the preparation of jellies. This alginate absorbs water quickly, which make this property has been highly useful in various industries like paint industries, textile industries, paper industries, and this particular alginate is also being used in the production of this toothpaste, soaps, etc. And also it has been used as waterproofs because they have an ability of absorbing the water. Hence it has been used in waterproofing, fireproofing, fabrics, and also gelling agent. It is also been used in cosmetics, etc. And also apart from this brown algae producing this alginate, there are certain bacteria also has an ability of producing this alginic acid. That is notably one of the species, soil bacteria, free living free living nitrogen fixing bacteria called as azotobacter has an ability of producing this alginic acid. Hence, to conclude, this sargassum is one of a free floating, the dominant species. Most of the species are free floating marine algae, and few of them are also found to attach onto the substratum. And the entire life cycle, the diploid phase dominates. That is, the thallus is diploid, where this particular thallus can be distinguished into the can be compared to that of a angiosperm, where angiosperm bushy plant, where they have a stem-like, root-like, and leaf-like structures. And another important thing is no asexual reproduction, that is the sporulation is completely lacking in this group of organism. And the vegetative reproduction is only by the process is by the process of fragmentation. Sexual reproduction is by oogamous, where the gametes are produced in the special structures called as conceptacles, where they produces anthrozoids and the oospheres or the egg cells as a gametes. Once they fuse, they form a diploid zygote. This zygote develops into a new individual, and this particular organism or this particular brown algae forms the algal blooms. And also they have been highly has got a high economic value because of the production of this alginic acid. So thank you, students, for listening to this particular talk, to this particular class. And if you have any doubts,